There's a strong rationale to combine monoclonal antibodies in frontline setting for transplant ineligible patients. The reason being, monoclonal antibodies generally tend to have minimal side effects, short term and both long term, and they generally do not overlap with the side effects with other drugs that you're thinking about combining them with. Um, you know, the best example for that would probably be the combination of daratumumab with VMP in the frontline setting. The ALCYONE trial, which read out at the end of 2017, it was presented as a late-breaking abstract and subsequently published in early parts of 2018, was a large randomized phase 3 trial done in transplant ineligible patients, where dara VMP was compared to VMP, which is a standard of care therapy in Europe. And the results showed better depth of response in the experimental arm, improvement in progression-free survival, which actually was the primary endpoint of the study, as well as improvement in the depth of response by MRD negativity, measured at 10 to the minus 5 with NGS method. And these results eventually led to the approval in the United States of this combination for frontline patients. From an efficacy standpoint, I've already shared the data. From a safety standpoint, in ALCYONE, there was a higher incidence of cytopenias and infections, specifically pneumonia. But it appeared that beyond that, there were no other safety signals that were observed in that combination. The POLLUX trial was a large randomized phase 3 trial that compared the combination of daratumumab Lendex with Lendex in patients who had at least one prior line of treatment. And this trial resulted back in 2016, leading to the approval of the combination of daratumumab Lendex in the first and beyond relapse setting. The interesting thing about the efficacy primary endpoint results were, at the time the trial read out, the median PFS on the dara Lendex arm had not been reached. More recently, late last year, we actually reported on those data, showing the median PFS being roughly a little over 44 months, which was quite impressive compared to what the median PFS had been reported for similar phase 3 studies comparing 3 drugs versus 2 drug combinations. One unique feature about the trial was both the arms continued therapy until relapse and progression, so it wasn't a fixed duration question being asked. There was almost a 4-fold increase in MRD negativity in the 3 drug combination arm compared to the 2 drug combination arm, and the overall response rate was roughly 93% versus 78%. Compared to the standard of care arm. The experience in the POLLUX trial set the stage up for a frontline trial with a similar design, looking at the combination of daratumumab Lendex versus Lendex for transplant ineligible patients. The study was designed in a 1:1 randomization to experimental versus standard of care arm. The treatment was continued until relapse or progression in both the arms. The primary endpoint for the study was median PFS. Important secondary endpoints were overall response rates, MRD negativity rates, as well as overall survival and duration of therapy. What was interesting to see about this trial as well is the standard of care arm performed quite well. The median PFS was 31.9 months on the standard of care arm. But despite that, the experimental arm continued to do well, reaching its primary endpoint, and this trial was presented as a late-breaking abstract at ASH in 2018. The overall response rates were again quite impressive in the mid-90s. MRD negativity rates were again almost 4-fold compared to the standard of care arm. When looking at the safety signal, nothing new was observed compared to what had been seen in the POLLUX experience. Cytopenias again, and a slightly higher rate of infections was observed in the dara-lendex arm on the MAIA trial. It's also important to note that the complete response or better rates were almost doubled in the experimental arm, 47.6% versus a little over 24% in the standard of care arm. The median follow-up at the time the study was presented was 28 months. So it's a little too early to see a difference in overall survival. And that's why the hazard ratio was about 0.78 at this follow-up time point. The assessment of minimal residual disease has been readily incorporated in clinical trials over the past 5 years in the early relapse setting as well as in the frontline setting. And it has been correlated with overall survival benefit. So achieving MRD negativity from a prognostic standpoint helps us in identifying patients who are going to do exceptionally well with any given therapy. At this point, we're not utilizing MRD results to modify treatment, but it does help inform us on what would be the best strategy to get patients to MRD negativity when they're sitting in front of you and having that discussion. So MAIA is one of the first frontline trials that has incorporated MRD assessments. ALCYONE was another frontline trial that actually incorporated MRD in the frontline setting. So these results are showing us that addition of a monoclonal antibody to available regimens improves the depth of response for patients.